So good afternoon, everyone. Thank you, Dr. Shrestha, for the opportunity to talk today. It's nice to be in front of all of you. Usually I sit in the back, so I'm on the other side today. As mentioned, my name is Nikki Armstrong. I'm an assistant professor in the department here in epidemiology. So today I wanted to talk a little bit about some of my research interests, which lie generally in the field of cardiovascular and cardiometabolic genetic epidemiology. So before we start, I did want to mention, if you have any questions at all, please feel free to ask. I don't mind at all. Let's make this more of like a dialogue as opposed to a genetics lecture. You're in here with me for an hour or so. I would like it to be as entertaining as possible. So it was suggested for me to start out with a quick overview of my academic journey. So my background, give a quick overview of cardiovascular diseases, as well as health disparities of cardiovascular diseases, or these gaps in knowledge in cardiovascular disease research, which is sort of a hot topic right now in not the best of ways. And so ultimately, the goal is, how do we address both of these things using genetic epidemiology? So the back half of my talk will focus more on a couple of my research areas. The first one being pharmacogenomic analysis and risk prediction, and then predicting cardiometabolic risk in people living with HIV infection. So then after that, we can open up for more discussion or any questions that are remaining. Okay, so quick background, okay? Let me pull up my map. So a little bit about me. I was born in Connecticut, and later my family and I moved to a small farming town, if you will, in Northwest Ohio. I did my undergrad, as Dr. Shrestha mentioned, at Miami University, where I studied microbiology. So it was during my junior year where I fell in love with research and started thinking more about a research career, graduate schools, things along that nature. So I found a master's program at East Carolina University, which is on the eastern side of North Carolina, in their microbiology and immunology department. Okay. So I went to ECU originally in 2012 to work on flu vaccination research. This is what I was doing in undergrad, really enjoyed it. Wanted to, have a career out of that. However, I rotated in a cancer biology lab and decided to do my thesis work on that. So what brought me to ECU is something that I didn't even do. While I was doing cell signaling work in cancer, so this was breast cancer, liver cancer, and prostate cancer, one of my advisors at the time was a doctoral advisor, doctoral director actually, at one of the programs, and came to me and was like, hey, there's this new faculty member. He's looking for PhD students. You're looking for a PhD. Are you interested? And a lot of that, what he was studying, focused on population genetics of chronic disease, things like big data sets. And I was like, sure, all of those things sound great. I really have no idea what any of that means in that context, but I like data, and big data has to be, better than small data, right? So yeah, that sounds great. So I did my dissertation there, focusing on multi-omic markers of recurrent stroke. So in 2018, I was presenting some of my work at an international stroke genetics workshop, where I met one of the faculty members in the department. Not gonna highlight, but Dr. Irvin, sorry, threw you under the bus there. But so I met Dr. Irvin Ryan there, and I kind of really just sort of lucked into a postdoc position here. Now I'm at UAB. Okay. And so I was in this postdoc position. It was a 3-year postdoc. The majority of it was during COVID. I then took an instructor position, and then my current position started in 2023, which is an assistant professor position. So technically, I've been here at UAB in some capacity since middle of 2019. My academic journey overall, to sum it up, has really been shaped by a mix of luck, preparedness, how they say preparedness favor, or luck favors a prepared, really. So preparedness and openness to new ideas. So this is kind of my little spiel about my academic journey. So I started with a clear focus in microbiology, right? It's what I really wanted to do. And then I had a whole bunch of unexpected opportunities. I rotated in a cancer biology lab, which I never thought I would do. I met with a trusted advisor who was like, hey, there's this new guy, you should go join his lab. And I was like, sure, that sounds great. I had a chance meeting at a conference that really worked out for me. And so it led me to new disciplines and ultimately shaped my career to what it is today. So all of that spiel, basically to say, embrace opportunities, focus on what you want to do, but be open to things as they come about. So now on to the science. All right, so cardiovascular disease, we've had infectious disease talks the last few times, but today we're going back to chronic and CVD. CVD is this large umbrella term for different diseases that affect heart and blood vessels. So my research commonly focuses on a few cardiovascular diseases, primarily stroke, myocardial infarction, so MI, heart attack, and heart failure, as well as risk factors. So those are typically cardiometabolic, so down on the bottom, so hypertension and diabetes. Okay. So more than 800,000 people die each year in the US from CVD, so this is a leading cause of death independently. And interestingly, in 2020, both heart disease, can't go any further, but heart disease and stroke, which I think I can highlight here, actually had more mortality, increase in mortality from 2019. So this is an issue that really isn't getting any better as time goes on. Okay, so the chart on the left presents prevalence breakdown of cardiovascular disease by age and sex, with and without hypertension. So you'll see this a lot. And so this is from NHANES data from 2015 to 2018. So the prevalence of CVD comprising CHD, so coronary heart disease, heart failure, stroke, and hypertension in adults greater than 20 is about 49.2% overall, and it increases with age in both men and women, so males and females. So if you remove hypertension, the prevalence is 9.3% overall, but it still also increases with age. Okay, so hypertension is an important factor for CVD, and we'll talk a little bit about that here in a few slides. So there are also apparent geographic disparities in cardiovascular disease as well, as you can see on the map. All right, so the light blue is indicative of the highest mortality rate. So Alabama's in light blue, most of them, unfortunately, followed by green. And if you just focus on the Southeast region, you can see higher mortality rates, really kind of in that Southeast area. Okay, so there's a number of reasons for this that have been proposed. Socioeconomic and social determinants are one of them. Rurality is another. Okay. But we know that there are racial disparities in cardiovascular disease. And again, I know this is kind of a hot topic. We're going to talk about it today, particularly with stroke, as well as MI or fatal CHD. So we're going to go over these two really fast. But in non-Hispanic Black males and females, they had higher age-adjusted stroke rates as compared to any other racial ethnic groups in the US. And it's also true of MI and fatal CHD, okay, as you can see on the other one. So this is a schematic that was adapted from a think piece, an expert analysis from the American College of Cardiology, and it highlights major factors and for worse outcomes and future directions regarding cardiovascular disease disparities. Okay, so we know there are major reasons driving this, social factors being the number one cause, suboptimal risk stratification strategies, if you will, medication adherence. These are reasons for worse outcomes, and future directions, which is part of the reason of this talk, are down there in green, right? So novel treatment strategies and trying to collect diverse data collection. Okay. So the majority of my research focuses on ways to address this using genetics and genetic epidemiology. Okay. So now we're going to dive into the genetic part, right? So genetic epidemiology is relatively new, and it's a growing field, and it looks at how our genes and the environment work together to influence disease in populations. So since the Human Genome Project, which completed about 20 years ago or so at this point, we've been able to better map genes linked to diseases and study how genes interact with the environment. And also use other omics, so things like genomics, well, that would be genomics, proteomics, epigenomics, things along that nature. And ultimately, how do we improve public health with that? So in the long run, the goal is to take what we learned about genetic risk and help it, or use it to help people, right? That's the main goal. Why are we in public health if we're not interested in helping people? And there really are two main ways that this can happen. So the first is on the left. You can find new pathways that cause disease, and you can ultimately lead to new treatments or ways to prevent disease that would help everyone. So this is more of an agnostic approach. The second approach is more of a personalized or precision medicine way. how someone's gene and their genetic makeup make them more likely to get a disease. This could lead to different treatments or ways that we can manage the disease more effectively. So I'm going to give an example that kind of runs through the whole gamut, right, from the very beginning into clinical application, which I think is kind of important. Again, because the main reason is to help people. So to study the genetic factors of a disease, first, really in theory, the main goal is to see how much of the disease is caused by genetics. So this is called heritability, and it ultimately tells us what percentage of the difference in the disease or trait, so this could be things like height, it could be eye color, it could be disease risk, are due to genetic factors versus non-genetic factors, so environmental factors, essentially. So the CAD, so coronary artery disease, example is what I'll use here. And so CAD, I think at this point, most people know, it's a pretty serious condition. Arteries that supply blood to the heart narrow, can lead to a lot of problems. So chest pains, heart attacks, sudden cardiac death, not great things. And we know based on previous studies that the heritability generally is between 40 to 60%. So that means 40 to 60% of the risk of CAD generally comes from genetics. So we know that our genes do play a big role in who does get the disease. And these numbers vary by study. But typically it's in that area. Okay. So using different methods like GWAS, we'll talk about that here in a minute, researchers have found harmful mutations in at least 9 genes that increase the risk of coronary artery disease. And so these genes generally affect pathways leading to cholesterol, so LDL, cholesterol, triglycerides, lipoprotein metabolism, things along that line. So just really quickly, GWAS. This is not a new term for everyone. Everyone maybe has heard of GWAS. Anybody? Sort of familiar? Okay. So GWAS is genome-wide association study. Basically, it's an agnostic or this just very overview analysis of all of your genetics, typically on autosomes. Okay, so chromosome 1 through 22, you scan through your genome and you look for single variants. Okay, these are single nucleotide polymorphisms. You'll see genetic variants, any sort of combination of those. And you see if alleles on there. So if you remember, you've got 2 copies of everything at a position, you see if one of those alleles are more likely than the other to confer disease risk. Okay. And so this is hypothesis generating work, right? We kind of go in with maybe somewhat of a leaning of what we think could be associated, but we have no real hypotheses that this specific variant is associated with CAD or hypertension. And so we're gaining information with that. Okay. So that was a little GWAS. Back to the CAD. Okay. So what's really exciting about the work that they did in CAD is that there are medications that are developed or already in use that can mimic protective effects of certain genes and genetic variants. So I'm sure most of you have probably heard of PCSK9 inhibitors. That mimics a genetic variant that lowers LDL, okay, by about 88%. Okay, so instead of having that genetic variant, we're mimicking that with this treatment. Okay. So it's kind of exciting. Continuing on with the CAD kind of topic before we get into my research, there was a study in 2019 by Kara et al. That created a polygenic risk score. And you'll hear a lot about those, and there will be more information on that later. But this was used from the UK Biobank, which is a very large data set. And this includes over 6 million variants in this risk score. So really, they wanted to see if this score, which really aggregates the effects of a lot of variants into one, and like I said, we'll go over that here in a minute, is similar. Okay, so can I identify people at high risk for CAD, similar to those people that have a mutation in one of 3 genes, LDLR, ApoB, and PCSK9. So it's a familial hypercholesterolemia mutation that it predicts about the same as that genetic variant. So they found that about 8%, so this is over in the distribution, about 8% of people who inherited this genetic predisposition conferred at least a 3-fold risk. And you can see that in the light blue. And then about 2.3% of the population in this like middle blue color right there had a 4-fold risk. And then about 0.5% had a 5-fold increased risk of CAD, just based on this genetic risk score. Okay. So what we're doing is we're stratifying people at highest risk. So here's one of the main limitations to, I guess, genetic epidemiology as a whole, okay? The whole field. Well, polygenic scores like the one created by Kara et al. That we just talked about for coronary artery disease, they can help identify people at highest risk. But these scores are often derived from a very non-diverse population, okay? Predominantly European populations. Okay. So the GWAS catalog is this publicly available resource. Researchers can upload their summary statistics there for various traits and variants. And then you can pull that down. You can use it for your analysis, so on and so forth. So it's all about sharing. Okay. So currently, over half the GWAS catalog consists of European studies, about 20% of it from Asian populations, so including both East and South Asian populations. And non-Asian studies, including those from African, Hispanic, and other admixed populations, make up about 18%. And you can see that. I don't know if you can see. Let's give my pointer up here, laser pointer. You can see it here in that yellow. Okay. So what's even more concerning to me is the aspect of underrepresentation of individuals. So that was on studies. This is on individuals. So 78, almost 79% of all the individuals in the GWAS catalog are from European ancestral populations, about 10% of Asian populations, and about 3% of non-European populations. Okay, so this highlights the need for more inclusive studies, particularly for diseases that disproportionately affect non-European populations. Okay. We good so far? Anybody have any questions? Burning questions? Okay, so we'll continue. This is going a little bit more into my research. So we talked about GWAS, genome-wide association studies, again, typically hypothesis generating scans across the genome, and it aims to identify genetic markers associated with a specific disease of interest. However, in addition to outcome-based GWAS, so like, does someone have a stroke or not? Does someone have hypertension or not? You can also do pharmacogenomic analyses, which can provide information regarding genetic predisposition and susceptibility to different treatments. And that's what I want to spend the first part of this talk on, with two separate projects, kind of leading into each other. Okay. So one of the first projects that I worked on, developed, if you will, as a postdoc, was identifying genetic contributors to antihypertensive efficacy in GenHat. So you may have heard a little bit about AllHat or GenHat, but briefly, we're going to start with AllHat because that's the parent study. AllHat is the antihypertensive and lipid-lowering treatment to prevent heart attack trial. Okay. And so AllHat enrolled over 42,000 participants. Everyone was a little older. They were 55 years or older. Everybody had hypertension, and everyone had at least one additional cardiovascular disease risk factor. Okay, so that could be diabetes, could be several different things. Okay. The trial compared effects of 3 different blood pressure lowering drugs, okay, different classes. There was a calcium channel blocker, an ACE inhibitor, which we'll talk about a little bit later, and an alpha adrenergic blocker. Okay, and so we're comparing those. They were comparing those with the effects of a thiazide-like diuretic. Okay, and so that was the control. All right, and so there were a number of different cardiovascular outcomes. GenHat is an ancillary study to AllHat. Okay, it was a pharmacogenomic study. It aimed to see if variation in hypertension susceptibility genes interacted with blood pressure medications to affect cardiovascular outcomes. So those blood pressure medications were the 4 that I named before. There is a set of about 6,000, almost 7,000 individuals that have genome-wide microarray data that were collected. And so after quality control, there's about 6,900 participants who are Black, from the diuretic arm and the ACE inhibitor arm. So the diuretic that we used was chlorthalidone. The ACE inhibitor was lisinopril. And that's just a little flow chart of how we got there. Okay, so why did we do this, right? So this table, not part of my work, but it shows that blood pressure or hypertension cascade by both race and ethnicity. So there have been a number of studies that looked at this, the difference between hypertension control between races, Black, Asian, Hispanic adults compared to white adults. Okay. And in a 2021 study, which is where this came, Black Americans had a higher hypertension prevalence compared to white Americans. They had similar rates of awareness and treatment, but lower control rates, as you can see here. So these differences are often linked to things like healthcare systems, physician-related factors, but we also know that all patients do not respond the same way to treatment. And so pharmacogenomic studies help us understand how genetic variations affect drug response. Okay. So on the right is a summary of a handful of pharmacogenomic studies on blood pressure response, essentially from the last decade. There's not a whole lot of them. On the right is over here. So on the right is the number of individuals that comprise each study. Okay. The highlighted numbers in red show the small sample sizes of individuals who are of African descent or who are Black. And then to address this, we focus on identifying the genetic contribution to chlorthalidone efficacy in nearly 4,300 people. And then we had 570 in replication. So this was one of the largest, it was the largest study in this population for this drug. Okay. And again, this is one population, this is one drug. There's a lot of work in this space that needs to be done. And one of our more interesting findings, if not our most interesting finding, was a genetic locus on chromosome 5 related to diastolic blood pressure response. And so several variants in this region reach a genome-wide significance level. Okay. And these significance levels are usually a little bit higher. It takes into account, It takes into account multiple tests, right? So each variant essentially is its own statistical test. So these are p-values that are 5 times 10 to the negative 8, 5 times 10 to the negative 9, depending upon what you're studying, really. But they're very significant p-values, very small. And so you really want your genetic variants to reach that red threshold. This right here is called the Manhattan plot. I'm not sure if people are familiar with those, but it's one way to visualize GWAS results. And it's called the Manhattan plot because ideally what you want to see are these very tall lines that look like skyscrapers, right? The Manhattan skyline. Oh, we may or may not, depending upon what you see here, consider that to be a very tall skyscraper. But we'll call it a moderate skyscraper, right? Like it passed the red line. So you really want all of these variants in line there. On the x-axis is chromosomes, so 1 through 22. It labels to 21, but that light green is 22. And then on the y is the negative log 10 p-value. Okay, so each dot is a single SNP. It's a p-value for that SNP, that association. Okay. All right, so these variants that we identified, okay, they were located upstream of a gene called CDH9. Okay, and this encodes for a cadherin that's involved in cell adhesion. All right, and so not to kind of go down a rabbit hole, but a lot of these variants have been linked to coronary artery calcification and serve as reliable cell surface markers for renal fibroblasts. So these are all, pretty interesting results that may play a role in blood pressure regulation. We know those are all sort of related. Okay. So when examining the lead variant or most significant variant in the region, we observed a dose-dependent blood pressure, diastolic blood pressure response to the number of affected alleles. And in this case, we are looking at the number of Gs. Okay, so in the first column over here, right here, you have 0 G alleles, and then you have 1, and then you have 2. So individuals with 2 copies of the G allele had the best diastolic blood pressure response to chlorthalidone. So there was an average reduction of about 4 millimeters of mercury. When you had 1 copy of the G allele, you had about a 3.6 millimeters of mercury diastolic blood pressure change, and then about 2.5 if you didn't have any G alleles. Okay. All right. So how does this work? So you have all these p-values, you have all of this data. Are you going to test everyone for one specific variant? Are you going to be like, well, you have a G allele here. Like, what does that actually mean? So using these genome-wide scans, we can now predict genetic risk through things like polygenic risk scores, or PRS, you'll see. So polygenic risk score is this tool that is used to predict an individual risk for a particular disease or trait. And it's based on a combined effect of multiple genetic variants. So each one of these variants confers a very small amount to the total risk we are identifying. But when you add them all together, these risks essentially are cumulative. And we know that this works because we know that CVDs are polygenic, right? So multiple genes are affecting cardiovascular disease. There may be monogenic causes, but for the most part, it's this mix of a whole bunch of different genes. So because these are polygenic, these scores are quite powerful or potentially powerful in identifying people who are at high risk for certain things. So very briefly, a PRS is built from GWAS summary statistics, why it's important to have GWAS statistics that are large scale, that have diverse makeup. And then the optimization step involves using a training cohort to select essentially the best method or the best parameters based on essentially the genetic makeup of the specific disease. So that genetic architecture. Validation occurs, which is kind of that middle step there on a completely different cohort, an independent cohort. And it really is essential to making sure that what you have shown that performance is in fact reliable. And then finally, you can start adding things to make a composite score. So things like demographics or clinical factors, lifestyle factors, and that helps for a more personalized risk prediction. So something that is interesting about PRS, it's a genetic score, it's a germline score. So your germline genetics are your germline genetics, right? Like from conception at birth. And so you can use these score, not that I suggest like going and, getting a genetic test, for like a 5-month-old, but you could conceivably. And you can see the risk that your genetics confer throughout your life. Okay. So why, again, are these appealing, right? Because they can be clinically applied to personalized screening. You can also use them for prevention strategies. So once a PRS is generated, optimized, validated, it goes through all of the steps. An individual's score can determine their position within a population's risk distribution. So again, these are normally distributed. You have tails, you've got a big middle. And so, for example, if someone has a low genetic risk, so we can say those are the people who are in green for whatever disease X, for example, their lifestyle factors will help determine their approach to screening or prevention, right? So if you live a generally healthy life, you're taking care, you exercise, things along that nature, you may not need any additional intervention for disease X. Similarly, say you have a couple tests come back, you're a little elevated, your blood pressure is a little high, HbA1c might be a little high, things along that nature. Maybe these people would benefit from an early intervention, okay, based on their genetic risk. Similarly, individuals with high genetic risk, so those are the people in the red, they may be at high genetic risk, but if you have like a more healthier lifestyle, you may not have any sort of intervention, or you may have an early intervention that is similar to the one that you would give to somebody at low genetic risk. And then if you're at high genetic risk and you have high clinical profile, you might need aggressive intervention. Okay, and it's all based on your genetics. All right, so another resource, if you're interested in this kind of work, is the PGS catalog or polygenic risk score catalogs. Currently, PRS scores are being developed, but they might not necessarily be developed for all ancestral groups at the same rate, mainly because there isn't enough GWAS data for these groups, right? So it all kind of goes back to that first step in making sure that, these studies focus on everyone from the beginning. So this is a significant barrier when you're generating polygenic risk scores. Out of curiosity, kind of went back, checked the PGS catalog. So this was as yesterday. So I have updated this from the last talk where I've kind of talked about this. But as of yesterday, the catalog included over 650 traits. There are more than 5,000 polygenic risk scores. However, when you search for scores based on just, say, African ancestry specific data, so that's the summary statistics, right? Not multi-ancestral, which is a different kind of a deal, but just African ancestry, there's only about 40 PRSs, and of those, only about 6 are fully polygenic, meaning they scan the genome. They're not just specific 6 variant scores, 10 variant scores, 40 variant scores. Okay, and a lot of those are for things like LDL, height, smoking, asthma, BMI, and HDL was the other one. Okay, so that was your 6. All right, so others seem, again, to just kind of rely on a few significant variants, and that's okay as well. But if you're really looking for this polygenic nature, I think we need to kind of bolster this up a little bit. Now, a little caveat, there may be more scores that exist and they're just not in this catalog. Okay, so it might not be as doom and gloomy as I just kind of made it to seem, but they aren't available readily yet. Maybe they're working on that. Again, this is a problem because if we do want to include those scores in everyday healthcare, we are leaving out populations that could benefit the most from early interventions and these tools. Okay. So one way, I kind of mentioned it before, or at least alluded to it, that we are trying to remedy this is by evaluating transferability of scores. So these could be multi-ancestral, these could be generated in European ancestries, and then applying them to other ancestral groups. And so what we have found that prediction accuracy relative to European, so European score versus a European polygenic risk score, they transfer very well, like they help predict very well. This violin plot shows that admixed American scores, South Asian, East Asian, and African scores do not predict as well. Okay, so there's only about 60% in the admixed population and about 25% prediction accuracy relative to white and African. Okay, so back to our GenHat example. So we talked a little bit about the GWAS. That was a project, and then we kind of thought about it, well, what does that actually tell us? Is there a way that we could take this information and make it relevant to the clinic? And so we, or at least in theory, relevant, potentially relevant. So back to our pharmacogenomic work, we were curious if a general systolic blood pressure, so this has nothing to do with pharmacogenomics, this is just a blood pressure score for systolic blood pressure, would be associated with differences in response to hypertension treatment. So can a systolic blood pressure score tell you how well you will react to a certain drug? Okay. So again, this was in GenHat. It was conducted using about 6,000 Black participants with genetics data. It was randomized. We did 2 different studies, one with the chlorthalidone arm only and lisinopril arm only. So the chlorthalidone arm was about 3,700. And this is the demographics table of just the chlorthalidone arm. Okay, so the lisinopril arm is not up here. Briefly, on average, participants in this arm had an elevated BMI, so about 30, mild reduction in kidney function, so EGFR of about 81. About 41% had a baseline diagnosis of diabetes. And remember, I did tell you, like, to be in ALLHAT, you had to have another CVD risk factor. So a lot of that was diabetes. So diabetes was elevated, and obviously so was baseline blood pressure because everyone was had hypertension. Okay, so we used 2 polygenic risk scores from the PGS catalog. We generated genetic risk scores for each person in this group in GenHat based on their genome-wide score. And that score was normally distributed. We then divided that score into 5 groups, so quintiles. Each one being about 20% of the score. So the people, if you think about the distribution, the bell curve, everyone over here on the right tail, we're in the highest risk, 20%. In the bottom tail would be the lowest risk. So that would be the highest genetic risk for elevated blood pressure, or the lowest genetic risk for elevated blood pressure, systolic blood pressure. Okay. All right. And so that was based on previous work. That's where we got those thresholds. And so among GenHat participants taking chlorthalidone, each increasing quintile of the primary score was associated with decreased 6-month systolic blood pressure response. Okay. So those in the lowest quintile, so that would be Q1. Okay. So these are the people that genetically are at least risk of having elevated systolic blood pressure. I know it takes a second to think about it. So those are the people genetically that are probably most unlikely to have systolic hypertension. Okay. They had a mean blood pressure change, systolic blood pressure change, sorry, of about 10 millimeters of mercury over 6 months. So that is not an insignificant change. versus a mean blood pressure change of about 6.5 in Q3. I have my pointer. Like I really need a stick here. Q3 is our median. So we're comparing everything to Q3, right? You don't want to do extremes like the people at the highest versus the people at the lowest. You want to do the people at the highest and the lowest versus the people in the middle. So like more of a normal situation. And then the people with the genetically highest risk of having elevated systolic blood pressure had a reduction of about 5 millimeters of mercury, which again, I mean, it's not terrible, but compared to these people at the highest or at the lowest risk, it was a difference. And so this was also reflected in the other polygenic risk score, which is in the screen box here, that we utilized. So we did 2 scores and we replicated both internally. So really it was kind of like a 4 score deal, and it replicated there. The effects might not have been as large, but it's still about 9 millimeters of mercury, ranging between 5 and 9. Okay. However, we did not observe any difference of this systolic polygenic risk score on the change in diastolic blood pressure. So using a systolic PRS to evaluate diastolic blood pressure wasn't really there. We also examined these scores on the lisinopril group, and we did not observe any differences in that either. We could talk about that in a second. So up to this point, we've been looking at how well individuals responded to treatment, but we also wanted to see if there were anybody or if there was anything that had to do with antihypertensive non-response. Okay, so this led us to investigate ATRH, which is apparent treatment-resistant hypertension. So resistant hypertension overall is defined as blood pressure above target while using 3 or more antihypertensive classes, right? So it could be well, it usually is a diuretic. So a diuretic and things like an ASI, a beta blocker, an alpha blocker. Or you could have 4 or more drug classes with controlled blood pressure. So if you're on 4 drugs and you have controlled blood pressure, you would still be a case in this situation. Apparent treatment-resistant hypertension is commonly used in observational studies or studies similar to this, where you can't really rule out pseudo-resistance. And when I say pseudo-resistance, these are things like medication adherence. So we didn't have access to medication adherence, improper blood pressure measurement, or things like white coat effect. So if you've ever gone to the doctor and your blood pressure has gone like all the way up and you're like, I promise you it's not that high, that could be because you're scared of doctors, which is kind of like me. Anyway, so these are things that you can't rule out. So of the 6,000 total GenHap participants, there were about 1,700 that we could either classify as a case or a control. So there were 634 cases and about 1,000 controls, almost 1,100 controls. Okay, and these people were followed up through year 3. Okay, so this is over a 3-year follow-up. And we also used regards as a replication cohort for this section. So sure everyone has at least heard of regards a name, but just very briefly, national longitudinal cohort study, over 30,000 people were enrolled, Black and white participants across the contiguous US. Okay. Ultimately, they were looking to determine reasons underlying higher rates of stroke mortality among Black compared to white participants, as well as people who live in the stroke belt. So the southeast region of the US. Why did they have higher stroke rates? Why did they have worse death rates? So participants completed a telephone call, computer-assisted phone interview that helped collect different survey data, like questionnaires, as well as some demographic information. They also completed an in-home visit where things like blood pressure was measured, you got blood drawn, urine specimens were collected. And then participants have been contacted about 6-month intervals to obtain information about outcomes. So genotyping was performed on about a subset of 10,000 of these regards participants, predominantly more so in Black participants. About 80% of those were Black that were genotyped. All right. So again, that was a replication cohort, but we're going to talk about regards anyway. So a little regards information. So in GenHat, we observed a 1 standard deviation increase in the blood pressure PRS associated with a 40% increase in the odds of treatment-resistant hypertension. Okay, as you can see here on the bottom, the 1 standard deviation. I keep forgetting my mouse is a pointer. Okay, so this association was replicated in regards. Okay, so very similar results, both in Black and white participants of regards. Okay, so Gen had participants in the highest quintile if we were actually looking at categorical. So the highest quintile had a 67% increase in the odds of HERH compared to those in Q3, so that median or middle quintile. And then there was a significant association detected among people in Q1 compared to Q3. So these individuals had about a 37% lower odds of HERH compared to individuals in Q3. Okay. And again, that was shown in regards as well. So we also looked at model prediction. So if you add in this risk score, either as a quintile, which is on the side, or as a continuous variable per standard deviation, and you add things into the model that are clinical. Okay, so that would be like model C and model D, where you add in things like eGFR or body mass index, diabetes, smoking, prediction increases quite substantially. So it goes from about a prediction model of 70% to almost 75 or 76% with all of the factors. So everything together is about 76, 74 if you just add in the genetic data. It's about 75 if you add in the clinical variables. So this PRS was predicting about the same as these clinical variables. There wasn't a statistical difference between the two of them. Okay. So again, this is important because like you can get this information at any time in an individual's life. This germline data. Okay, so really the take-home messages here of this AHT, so antihypertensive pharmacogenomic analysis. We had both an association and a PRS component. So the association component is what are these genetic factors? Are there genetic factors that can help predict which patients are more likely to experience either beneficial or adverse responses to certain drugs? Chlorthalidone, lisinopril, whatever it may be. Ultimately, can we help personalize hypertension strategies a little bit better? But again, with these association where you get like these files that have millions and millions of variants, how do you do that? How do you pick just one? And polygenic risk scores have this ability to help translate that into a more clinical approach, or at least a more tangible thing. What we observed was a higher systolic blood pressure PRS. So greater genetic risk for elevated systolic blood pressure is associated with reduced systolic blood pressure response to chlorthalidone. Okay. So if you were in Q5, you had a reduction in your response. It wasn't as important. It wasn't as good. I guess good is probably the word, as if you were in Q1. Participants in Q1, so in the lowest quintile, experienced a greater reduction in systolic blood pressure compared to those in higher quintiles. And then this was only observed for systolic blood pressure in chlorthalidone, suggesting that this polygenic risk score is specific for chlorthalidone, not lisinopril. And that there also could be like some little caveats. It might have something to do with sample size, even though our sample size for lisinopril was about 2,000. So it wasn't a small set, but there may be some differences there. Maybe other risk scores affect lisinopril better, but this is what we observed in our data. Okay. Also, sorry, individuals with higher PRS had increased odds of developing treatment-resistant hypertension. Okay, so which is a more severe form of hypertension. Okay, and then just kind of switching things over to more current work. Okay, so this is the next part, and we're going to go through this pretty quickly so you have some time. I wanted to talk a little bit about other ways we can use polygenic risk scores. Okay, so this is looking at predicting cardiometabolic risk in individuals with HIV. And so there have been a number of different statistics. World Health Organization has estimated over 37 million people are currently living with HIV. And that might be a little bit dated. It might be a little bit more at this point. Nearly 2 million become affected every year. So antiretroviral treatment, which is ART or combination ART, has largely transformed HIV into a more chronic disease. And therefore, people are living longer, which is good, right? However, when you live longer, the risk for cardiovascular disease also increases. So you develop more chronic diseases in the interim. So there was a systematic review in 2018 that reported people living with HIV are 2.16 times more likely to have cardiovascular disease compared to those that do not have HIV. And that this is pretty similar to, so these are odds ratios, these are risk ratios, take that into account, but they're pretty similar to different factors that were identified in the inter-heart study. So things like hypertension, diabetes, lipids, and smoking. So the main question is it time to recognize HIV as maybe a risk factor for cardiovascular disease? Could it be? And then it's been reported that both viral infection as well as ART contribute to a number of different mechanisms biologically, things like endothelial dysfunction, which are precursor to atherosclerosis. So it does make biological sense there too. Okay, also inflammation, which we'll get to. So a lot of this work focused on using genetics data from regards, which we talked about before, as a non-HIV population, as well as CNICS, which is the CFAR Networks of Integrated Clinical Systems study. Okay, so CNICS is an electronic medical records network that's made up of currently 10 institutions across the country, and UAB is one of them. We could talk more about CNICS, but I'm not sure if anyone's heard of CNICS. It's a really great resource. I hadn't really heard about it until this project, along with another project, but it's quite extensive. It has a lot of follow-up time. There are over 400 peer-reviewed publications. There's a lot of funding. They have a vast data repository, which we can talk about. And they have a really good mentoring program, which has been very nice because they've been very welcoming and collaborative. When I kind of came in and was like, this is kind of what I want to do, but I don't really know what I'm doing. And so they've been very helpful. So if you want to know more about CNICS, let me know and we can talk about it, or I can guide you to people who know more about it than I do. So the main point and goal of this work is to evaluate the utility of a meta-PRS, which we'll talk about in a second, for incident MI and prevalent T2D in populations with and without HIV, okay, in both Black and white participants. And so these are our research hypotheses, but typically or generally, I guess, we want to know if a PRS generated in a non-HIV population will translate to help predict CVD in people with HIV. And you could think like, well, genetically, shouldn't the risk be the same? But it's also what part of the CVD is genetic and what part of it is related to environmental factors like inflammation, lipid metabolism, association of lipid metabolism with ART, things along that lines. Another question is transferability will be, or hypothesis, sorry, will be transferability will be greater for white compared to Black, and that meta PRS for both lipid and inflammatory markers will improve prediction. Okay, but we'll probably improve it more for HIV population. And so a meta PRS or a meta polygenic risk score is very similar to a polygenic risk score, right? It's used to estimate a person's risk for a disease or trait based on many small genetic variations. So similar to a PRS, okay, it adds up or it really kind of calculates a person's likelihood of a certain trait by combining multiple PRS scores, often from different studies, with related traits, right? So like we'll take a sports analogy. So like if you're predicting how well a person might be at basketball, right? You could be like, you could look at height score, right? But it might also take into account things like stamina or coordination, or speed, like are you agile? Like, can you see? Like these are all things that probably play a role in whether someone can play basketball or not. Same thing with like a cardiovascular meta PRS, right? So things like hypertension can go in, systolic blood pressure, which would also probably not work with hypertension, but diabetes, things along that line, all together into one score that ultimately creates a more powerful and accurate prediction in theory. So for this project, we're starting with single trait polygenic risk scores for from the PGS catalog. We wanted to create a meta PRS for lipids and a meta PRS for inflammation. And these are the different traits we were looking at. So for lipids, we had triglycerides, LDL, and HDL. For meta inflammation markers, we have things like IL-6, IL-8, CRP, things along the lines there. And then for each meta score, we also include the outcome score as well. So if we were looking for the meta PRS for lipids, for MI, you would have a 4-score score, if that makes sense. So you would have MI and then these 3 lipid markers all together to make one score. So it's essentially a meta-analysis of PRSs in a nutshell. Okay, so we wanted to use 70% of regards, so a larger data set. We split regards into two, 70 and 30. And that 70% of our regards genetic study is our testing data set. 30% of regards is a validation, with 100% of scenics being our primary validation study. And this is just a table presenting key demographics. Really, I just wanted to give you sample sizes. So look at the first row. For regards testing, there's about 6,000 Black, 1,100 whites. In scenics, there's about 4,100 Black, 3,400 whites. In validation for regards, anywhere between 400, about 500 and 2,500 people. Okay. So this is not a stats lecture. I am not Dr. Tawari. So I just wanted to kind of show you what this looks like a little bit. This is the meta PRS. Basically, again, what we're doing for inflammation, there's 6 polygenic risk scores that go into the score, and then you end up outputting 1. So you need things like the 0 mean unit standardized risk scores in the validation set, whichever one that would be. So that's those zetas. You've got beta values, which are univariate parameters from regards training, and correlation coefficients between the scores. Okay. And so what I'm going to show you today is association between the single trait PRSs or meta PRSs and outcomes. So no prediction today. We're still sort of working through that. I'm sort of working through that. So in scenics, what we ended up seeing is really no improvement for MI and Black participants of scenics between the single trait PRS and our 2 meta PRSs, inflammatory markers or lipids. For our white participants in scenics, we did see the meta inflammation score was about the same, these two right here. For the T2D Black, our meta lipid score was slightly better, slightly. I mean, the confidence intervals are overlapping. I mean, they're about the same. And then for white, both meta PRSs were in fact worse. When we looked at regards validation, so again, this is a score generated in regards, also validated in regards, and validated in scenics. We didn't see any improvement with the meta PRSs. So for myocardial infarction in both Black and white ancestries, none of the meta scores performed any better than single trait. For type 2 diabetes in Black and white ancestries, there was no clear improvement in performance either. Okay, so it didn't really, it didn't help any in regards. But it might have helped a little bit in scenics. And so some key points, the takeaways for the association. For Black participants, the meta PRS for T2D, specifically the meta lipids T2D PRS, showed a slight improvement. Meta PRS for MI showed no improvement. For white participants, both performed worse than single traits, suggesting that combining traits may not provide an advantage in predicting disease for white participants there. And it could have been, again, there's multiple reasons for that. In regards, there weren't any improvements for prediction for either PRS, for either outcome. So the single trait PRS seems sufficient for those diseases in the general population. Okay. So really, it may stem from a number of different things: genetic variants specific to population, those associated with lipid metabolism, which maybe the meta lipids can better capture. We know that there's an association between ART, and lipid metabolism, so maybe that's part of it. There might be a number of different things, environmental and lifestyle factors that we're kind of working through here. And then we're still going through the prediction part. So the main take-home message in the last 2 seconds, really, of the talk is just the importance of genetic epidemiology. What we're aiming to do, if you get anything out of the talk, just kind of knowing these parts. So unlocking disease mechanisms. We talked about GWAS and how they can identify genetic variants associated with diseases. This can ultimately help provide some insight to underlying biological pathways. Personalized medicine. These findings can lead to more tailored treatments and interventions based more on an individual's genetic risk. Risk prediction, so it could enhance accuracy of disease, leading to earlier interventions, which are important. And then ancestry-specific insights. So diverse populations enable more accurate and equitable risk assessments. And so with that, Thank you to my group who all helped with all of this data.